Hello, I'm Julia Murdock, and I'm doing this review of the progress check for Unit 6 for AP Biology for gene regulation. And this one question is, I think, a really good example of a question that has a lot of, it looks complex, but it really hits all the things that you want to understand for a lot of its unit. So I'm just going to read along with you here, okay? In mammals, you have the dark color of skin, hair, and eyes due to a pigment called melanin, the same one that you were doing with the rock pocket mice, right? Melanin is produced by special skin cells called melanocytes. The melanin is then transferred to other skin cells called keratinocytes. Melanocytes synthesize melanin using a multi-step metabolic pathway. The amount of melanin produced is dependent on the amount of enzymes TYR, TRP2, and TRP1. So here's the metabolic. We looked at this like Unit 2, remember, eons ago. Remember that? So you have one thing, tyrosine, and there's the enzyme that turns it into another thing, L-DOPA, and then another enzyme that turns it into yet another thing, which turns into another thing. So these enzymes are really key in the multi-step production of this stuff here, melanin. Melanin, which is going to make your skin a little darker and protect you from the sun and maybe protect you from skin cancer, right? That's the purpose of it. So metabolic pathway, 3 important enzymes that are needed to make the melanin. Okay, so if you go down here, now we have a synthesis pathway here. The peptide hormone, stimulating hormone, alpha MSH. There it is, a chemical signal. Here's a chemical signal pathway, just like from the hormone pathways we looked at before. It fits into a membrane MC1R, the same one you learned about in your HHMI activity, right? Which then stimulates adenylyl cyclase. You remember that, CAMP, and all of the cell signaling that comes through, and stimulates the genes, those 3 genes that make those important enzymes that create the melanin, okay? So there's that one picture that I've just decoded. Then we have more. It's a long question. Some mammals increase melanin production in response to UV radiation, okay? So it's the UV radiation that stimulates the whole process to begin with. So really, I almost think I would have put this part of the question first. Okay, here's the UV radiation. Here's the P53, and then you get the production of this chemical signal, the alpha MSH, that then, remember, is the thing that comes and gets those 3 enzymes going that are going to make the melanin. Everybody, I hope you're all with me. There's a lot going on here. Okay. So that's what stimulates the making of that signal that then goes and signals cells to make those 3 enzymes that then are in control of making the melanin. Okay, so now let's see what the question is asking at the end of all this. What is the point? Okay. Let me move this over here. There we go. Keep going down. Keep going down. There we go. Okay. So at the end of all this, it says, which of the following claims about TYR, TRP2, and TRP1 mammalian genes is most likely to be accurate? Okay, so we've got some choices here. Okay. The 3 genes are located next to each other on a single chromosome and are organized into an operon. That's your first choice. B. The 3 genes may be located on different chromosomes but are activated by the same transcription factor. There's B. The 3 genes are identical genes since they are activated by the same transcription factor. C. D. The 3 genes may be located on different chromosomes but with identical operator sequences. Okay. You should automatically eliminate A and D because those choices are talking about things that are only found in bacteria. And we are looking at mammals, right? Okay, so we don't have operons in the mammals, so not A. We don't have operator sequences in mammals, so not D. So it's got to be one of these 2. So let's see. Genes may be located on different chromosomes, activated by the same transcription factor. That's all eukaryotic. That makes sense. That could be it. Or the genes are identical genes. Now, is that possible if they're making 3 different enzymes? No. So the answer is B. Everybody with me on that? Okay, and that is actually the correct answer, B. Genes may be located on different chromosomes, but are activated by this, which makes sense, right? Because that one factor is going to wake up those 3 different genes because you need the melanin. Okay, let's go to the next one. Again, same exact thing, now different question. So now, all the same information, but now go down to the bottom and let's look at what they're asking. Which of the following claims best explains why keratinocytes do not produce melanin? Okay. Keratinocytes do not produce melanin. Okay. A. Keratinocytes do not contain the genes. B. Keratinocytes do not contain the MC1R gene. C. Keratinocytes do not express the MITF gene. D. Keratinocytes do not express the POMC gene. Okay. So we know A is wrong because every cell in the body is going to have the same genes. The genes are there. It's not like they're going to be missing. So A can't be right. And same for B. All the genes are there. So anything saying, oh, the gene isn't there, that's not right because we know the genes are all there. It's about whether they're being expressed or not. So it's C or D. Okay, so let's go up and just take a look at the information. Expressing MITF and expressing POMC and what would interfere with producing melanin. Okay. So, all right. Okay. The MITF is a transcription factor, not a gene. The POMC, gene, okay, produces that protein is then cleaved to produce alpha MSH. If you guys don't mind, I'm just going to open this up so we get an answer. Because I'm still on Florida time. Come on, just come down. There we go. Okay. Okay, there, yeah, okay, yeah. Okay, so MITF gene, and let's go to the explanation. There, okay. The MITF stands for melanocyte-inducing transcription factor, and its expression during embryonic development is critical to differentiation of melanocytes. The genes activated by M1TF include TYR, TRP, and TR1 genes that are involved in producing melanin. Okay, so what if we had put this? Okay, the keratinocytes do express this gene. UV radiation causes damage to DNA in keratinocytes, which activates the p53 protein. So that is expressed. Yeah, that's even complicated for me, guys. But at least you can get it down to 2. Right. Okay, let's go to the next one. All right, again, the same stuff. All the same question, just a different one down at the bottom, right? Oh, sorry, I didn't mean to give you the answer. There, let me just do that. Okay, so now they're asking, come on. Which of the following pieces of evidence would best support the researchers' claim? And their claim is that they're studying a mutant population of mice that don't show a tanning response. So they're not making melanin. Genetic testing showed that the pathway causing the production of it in response to UV radiation was fully functional. Thus, the researchers claimed that the lack of tanning response was due to an MC1R that wasn't working. The same as your HHMI activity. So let's go up here and I can show you why that's the answer. This one I understand. So if this isn't functioning, right, if it's the wrong shape, just like maybe in your mice, right, that means that if they bypass it somehow, they'll be able to prove that, which is what they do down here. So look at the choices. When researchers applied a drug that activates adenylyl cyclase bypassing that part, right? The level of melanin increased. The reason why that is actually the right answer is if you go back up here and look at the pathway, right? If this isn't working, but they add a chemical that activates the adenylyl cyclase, and they bypass this, and then they get melanin out of it, then that's proof. That's proof that this wasn't working, and if they bypass it, that supports the researcher's claim. Does that make sense? Yes? Okay. So then the other, if I just pull that in here, there, yeah. So when they apply a drug that activates it to the mice's ears, that must be fun. Correct. Activates it would increase the production of CAMP, which would activate protein kinases and just go down and then wind up making those 3 enzymes, which then control the making of the melanin. And the reason why the other ones you can eliminate is none of those go into supporting that pathway happening some other way. So when researchers viewed sections of mutant mouse ears, they found melanocyte numbers. That doesn't tell you how they got there or not. So that doesn't give you evidence. C, when researchers exposed the mutant mice to UV radiation, the amount of palm RNA in the keratinocytes didn't change. Again, that doesn't address the chemical signaling pathway and how you might bypass it. And when researchers exposed it to UV radiation, the level of melanin production didn't change. Again, that doesn't address, the one protein that's not working. Okay. We good? All right. 4. Again, the exact same question, but with a different question down at the end. Let's see what they're asking this time. Based on the information provided in Figure 1 and Figure 2, the same ones we've been looking at, which of the following best explains the effect of a mutation in the promoter of the TYR gene that prevents it from being transcribed? So if the TYR gene, one of the big enzymes that's in the metabolic pathway. Come on, just move. There. Okay. So if this guy that appears in 2 places is not made properly, right? The promoter region is mutated. The RNA polymerase can't bind and make that protein. That means that this entire metabolic pathway comes to a stop and no melanin can be made. You agree with me on this? Give me a thumbs up. Okay. Yeah, because if you don't have the first enzyme that changes the first molecules into the ones that these guys need to then change into the final molecules, you've stopped the process. So as a result, if you go down and look at the choices, now that you have that in your mind, which one would you pick? DNA damage due to UV radiation will be strongly inhibited. DNA damage due to UV radiation will be strongly inhibited. So, no, because no melanin is being made, so you wouldn't have any protection against UV radiation. So you'd eliminate A and B, right? No melanin, so all the damage is going to be allowed, not inhibited, right? So A and B are out. So it has to be C. Skin pigmentation will not change. Both of them say that. C and D say that. But they're for different reasons, so you got to pick the right reason. So resulting in a positive selection pressure or a negative selection pressure. Okay, so they're mixing in evolution here. So if you have mice that can't make melanin to protect themselves, are they going to be positively selected or negatively selected? I think negatively selected, right. So the answer should be D. Let's make sure Mrs. Murdock is right. Yes, thank goodness. Okay. Okay. All right, just going to do a couple, I think we're almost at the end here. Okay, so there's another one here with the same information. Come on, in, get out. There, okay. Different question. Which of the following best explains a process occurring between point 1 and point 2? This is basic, guys. This is right out of freshman year. Between the DNA here, what's happening between the DNA and then coming out of the nucleus and going to the ribosome, right? So you're looking for things that describe DNA, RNA editing. You're looking for RNA editing steps. That's it. It's as easy as that.